Hello, I am Kristiina Mäkinen from the University of Helsinki, Finland. One of the challenges in characterizing a particular protein complex is to determine its molecular composition. This requires development of methods to isolate and purify the complex from its cellular environment. The area of research in my group is plant virus host interactions, and therefore we are interested in the various viral ribonucleoprotein complexes formed during infection. In the past, we have successfully studied these complexes using affinity tag-based purification. In our hands, Twin-Strep-Tagged proved to be one of the best tags. We have, however, observed certain limitations in its use, one of them being low yield, and therefore we wanted to improve this method.
Hello, my name is Marta and I will guide you through the experimental protocol, but first let me give you an overview of the method. We employ Strep-Tactin macroprep beads to purify specific complexes between twin-strep-tagged proteins and their physiological binding partners. Strep-Tactin tetramer is shown in green, twin-strep-tagged protein in blue, its binding partner in orange, and 2 copies of the strep tag in red.